Please welcome to the stage Dr. Bill Andrews.
Thank you very much. Thank you very much. I do got to qualify that the first patient was not treated in November 2019. And as you'll hear during my talk, this gets delayed and delayed and delayed. I am really grateful to be here. I want to thank Al Sears, and he was saying how great a team he has here. I've been very impressed with the team, so thank you everybody. I can't list everybody's names. I am, because of time, I'm going to try to go fast through the fluff and focus more on the more important stuff. So let me just go ahead and get started. First, people ask why I'm so obsessed with curing aging. And this is a scene from Star Trek First Contact. This is when humans first make first contact with an alien species. Well, when that happens, I want to be there. I'm not afraid of dying. I'm afraid of missing out. And that's what, there's a lot of great things that are going to happen. I just want to be there for all of them. So let me, I'm pretty much telling you who I am. This is part of who I am. But let me just go through a few things. Austin already said most of what I'm about to say, but I'm a medical researcher, PhD in molecular and population genetics, which pretty much means I'm molecular and evolutionary biology. I've been in biotech for 38 years. Most of my focus has been on cancer and aging. I've done a lot of cancer research. In fact, I was second place National Inventor of the Year, which Austin just mentioned, for my cancer research. I'm featured in 2 documentaries, one called The Immortalist, one called The High. Immortalist almost won an Oscar. I highly recommend you watch it. It's about my research, and I co-star with Aubrey de Grey in that. And I'm an author of 2 books, Curing Aging and Telomere Lengthening, which I highly recommend. Easy to read books, really answers most of the questions that people ask me during my talks. Now, you're going to find that the things you have in your books are different from what I have here. That's because I've been in the news a lot lately. I've had to make some changes to my slide to address some issues that are being misunderstood. One is I've been involved in a lot of clinical studies. I've been the inventor of most of these things, and also, one of the inventors of them, and also involved in the clinical studies. That includes human growth hormone, which was approved in 1985, tissue plasminogen activator, approved in 1987, erythropoietin, the drug that Lance Armstrong used to cheat, approved in 1989, thrombomodulin, a really good drug to prevent blood clots, but the FDA withdrew it, and now it's back underway again, so still not approved. Beta-seron, the very first drug for treating multiple sclerosis, approved in 1993. And then telomerase, and mostly the clinical studies I've been involved with telomerase so far have been anti-cancer things, and those are still underway. But I want to make a really important message here. Every one of these studies involved a first-in-human stage, where the drug had never been tested in humans before, and now the first human was going to be done. Every single time this happened, there were critics all over the place saying, more studies are needed. You need to test and prove them in animals before you can actually test it in humans. You've got to be safer. But the fact is that, we're animals. The animals being tested are animals, but we are very different from any other animal. And we will never know if a drug's been tested and proven until we actually test it in humans. And so we can do animal tests all day long, or years and years and years, but it's not tested and proven until we test it in humans. And that step has to take place. There has to be a first in human, and there's always critics against it. I'm a member of 2 organizations that I'm very proud of. One is the Coalition for Radical Life Extension, and the other is the Transhumanist Party. I'm not only interested in extending lifespan, but enhancing lifespan as much as I can. But the real key word here is radical. I am very interested in radical life extension. Okay, now you hear all the time about people having cures for aging, but what they're talking about is they've had some biomarkers of aging that have been reversed. They haven't necessarily really cured aging. And so my opinion of curing aging is a little more radical. And so instead of having biomarkers of aging, I want to ask everybody one question. Is there anybody here who doesn't know which photo of Betty White was taken first? I don't think we need biomarkers. Well, we do for some of the studies, but I think biomarkers are important. And in studies that I'm doing, we are doing every biomarker anybody's ever thought of. But this is what's important to me, because I think that's what aging is. I can't put my finger on it. There's something different about her, a few wrinkles and stuff, but more than that, there's something different about her. But I don't think we need a biomarker to know it, okay? It's really obvious. And this is what I consider the cure for aging. I mean, when she gets cured, she's still going to age, so some people say it's not really a cure, but I'm not an expert in semantics. I call this cure for aging. And so this is what I call radical. This is radical life extension. That's what I want to see all of us do, and I call this the Betty White test. I believe that nobody is going to have a cure for aging until somebody passes, and so some type of treatment passes the Betty White test. So now, how am I going to try to do this? Okay, well, I believe that a kingpin of aging, not the only one, I think a kingpin of aging is the telomere. I think it's a very, very important thing that we've got to look at. No matter what else we do to cure aging, we'll never cure aging unless we also cure this. It might not be the only thing, but we have to cure this problem. So what is a telomere? Just in case there's anybody in the audience that hasn't heard me talk before, I'm going to quickly go through this and skip a lot of the fluff, as I said. Telomeres are very small things inside. We need to zoom in on a human being. We see that a human being is made up of a lot of cells, 100 trillion approximately, rounded off a lot. The cells contain nuclei, and inside the nuclei are found chromosomes. The chromosomes are made up of two DNA molecules. The top part and the bottom part, DNA and protein, but focus on the fact that there's a DNA molecule in there that's a really long string of beads, about 100 million beads in length. The beads are called bases, and this is where all your genes are that give you your hair color, your eye color, etc. Think of this long string of beads that's wrapped up like a slinky inside this chromosome. Think of it like a long shoelace. And at the tips of your shoelaces are the aglets that protect your shoelaces. Well, our DNA and our chromosomes have the same thing, shown here in yellow, and that's what a telomere is. So telomeres are the aglets of our chromosomes. Well, let's unravel the slinky that I mentioned of one of the telomeres. And we see that a telomere is about 15,000 bases in length, which is a very small part of the 100 million bases of the entire chromosome. So 15,000 bases, at least when we're first conceived. Okay, so here's why I'm in business. This is what I do. This is where all the problems begin. Because when we are that single cell embryo and our cells start to divide, each and every time our cells divide, the telomere gets a little shorter. So there's a lot of cell division required to go from a single cell embryo to a newborn baby. So during that process of just becoming a baby, your telomeres are already shortened a lot. So they were starting at 15,000 bases. When you're born, they're down to 10,000 bases. That's okay. That's like cutting 1/3 of the aglet off your shoelace. Your shoelace is fine. But the problem is it doesn't end there. You still have a lot more cell division, a lot of wounds to heal, a lot of immune things to fight, et cetera. So a lot of cell division going on. You're growing up, and the more cell division on and on and on, the shorter your telomeres get. When your telomeres get down to 5,000 bases, you die of old age. This is something that's not a theory anymore. There's every lab in the world working on cells in a petri dish know that when you culture cells, they can only divide a certain number of times, and they go into a stage called senescence, and shortly thereafter that, your cells die. And that's a limit to our lifespans. There's a theoretical maximum lifespan based on the rate that telomeres shorten of 125 years. And that's only if you have the perfect genetics and lead the perfect lifestyle. All the rest of us, our telomeres will shorten even faster, and I could spend an hour talking about that, but let me just review one more time. We're conceived at 15,000 bases, we're born at 10,000 bases, and we die of old age at 5,000 bases. This is what my life is all about, trying to fix this problem. Okay, so there's a lot of diseases that have now been shown to be affected by the length of your telomere. In fact, I don't even know of a disease anymore that hasn't been published in scientific peer-reviewed journals to not be affected by the length of your telomeres. Practically every disease we have could possibly be controlled if we could figure out a way to prevent this telomere shortening and actually reverse the telomere shortening. So how do we do this? How do we keep telomeres from shortening? Well, back in the 1990s, when I first got involved in all this, it was apparent that, I said that every time a cell divides, our telomeres get shorter. Well, they don't in our reproductive cells. In our reproductive cells, the telomeres don't get shorter. And that's important because if they did, our children will be born with shorter telomeres than we have. Their children will be born with shorter telomeres than they have. And we would have been extinct as a species a million years ago, before we were even humans. So our reproductive cells are doing something to prevent the telomere shortening. And in my research, I led the team that discovered this enzyme called telomerase, shown as it's a cartoon, the green squiggly thing is the DNA, now shown as a double helix. That factory-looking thing is this enzyme telomerase that binds to the tips of the chromosomes and lengthens the telomeres. In our reproductive cells, every time the cells divide, the telomeres still shorten, but then telomerase re-lengthens them. So it's a constant tug-of-war that's going on inside of our reproductive cells. So, why don't all of our cells produce telomerase? Why is it only our reproductive cells produce this enzyme telomerase? This is all related to gene regulation. And so what I want to mention, I said that our DNAs, where our genes are, they give us our hair color and eye color. This is an example of one gene that's along this long string of beads called the telomerase gene. Well, next to every gene is a regulatory element. This is what turns genes on and off. Well, in our reproductive cells, the gene is turned on. And so as a result, this gene is producing telomerase, and the telomerase lengthens our telomeres. But in all the other cells of our body, for some reason, there's this protein that binds to that regulatory element and shuts it off. It's called a repressor. Okay, so there's two ways to fix this problem. We don't want that protein shutting our gene off, and I could spend an hour explaining why in the world would we have evolved a mechanism like that that intentionally causes us to age. But there's actually, because of my PhD in evolution, I actually can really give a really good explanation for that if I had more time. I'm not going to do that. There's two ways to fix this problem. One is gene induction. If we could find something like a pill, like this little green thing, and you already know Alceres has something that does this, it dislodges the repressor and causes the gene to turn on. But I want you to think of the switch now as a dimmer switch, so it doesn't turn it all the way on. We're lucky to have some things that will turn it on partially, which is better than nothing, okay? At least it will slow down the aging process, or at least slow down the rate of telomere shortening a little bit. So doing that research is a lot of work, okay? So there's a lot of different departments in my company that do a lot of work to prepare things. Then we test things in a high-throughput robotic screening system. And then after we find things that actually partially do it, or something like maybe does a little or a lot, we then do more of all the same things again to design even better molecules to run back through the system again. So this process is going on and on and on all the time. We've now tested 350,000 different chemicals looking for things that will actually do this. We found 900 plus inducers that are found in 39 different families of chemicals. Now, the most potent that we have found to date is not enough to lengthen the telomeres as much as we see in our reproductive cells. The most potent chemical that we've found to date produces about as much telomerase, about 16% as much telomerase as is needed to actually be equivalent to what we see in our reproductive cells. So we have a lot more work to do. And the real big problem with this work, it costs us about $1 million a month to do. This is a very expensive research. All those departments I showed, all the robotic screening, retesting, things like that, costs us about $1 million a month to do. But when we have a treatment, those chemicals only cost like less than $1 per person to be able to take per dose. But that's the cost to my company, not the marketers guarantee will increase the price a lot. But my cost to produce enough to give one person one dose would only be like less than $1 per person. Okay, so that's one way. Okay, that's one of the main ways we're doing it. Another way is called gene delivery, also known as gene therapy. And that's what Austin was mentioning that we're actually doing clinical studies on right now. I want to just basically go through what gene therapy is. I want you to think of a human cell as a soap bubble. Okay? Then there's another little soap bubble that's smaller, and this is called the vehicle. It's actually the gene therapy. Some people call it a virus, some people call it a vector. A vehicle is a name that I prefer. And inside this vehicle, we put the gene for telomerase, or any other gene, depending on what you want to deliver. Gene therapy is a method of delivering genes to cells. In our case, we are delivering the telomerase gene to cells. So anybody who's blown soap bubbles before knows that when two soap bubbles come together, they fuse. And so this is what happens. When we inject a gene therapy into the body, these little bubbles fuse with your cells that are bubbles, and the gene gets inserted into your cell. That's how it works. Once the gene gets in there, it starts to produce telomerase. And what's really nice about gene therapy is it produces a lot of telomerase. So we actually can produce, right at the moment, 30 times more telomerase in a human cell using gene therapy than our reproductive cells do. So we produce 30 times more telomerase in a human cell than is needed to actually stop telomere shortening, and it actually lengthens the telomeres. It's so potent. We and others, labs elsewhere in the world, have done a lot of reversing of aging in human cells. In vitro, using this by every method of measurement imaginable. We've actually turned old human cells in a petri dish into young cells. People have also done it with human skin grown on the back of a mouse using the therapy. They have seen old skin become young again in every way imaginable when human skin is grown on the back of a mouse. And then people have taken engineered mice. And I want to say engineered mice because mice actually don't age like humans. There's a lot of studies you hear about and stuff like that, but most labs actually find that mice don't have telomere shortening. Their aging is very different from humans. But people have developed engineered mice that can be used in the studies, and they have shown that they've been able to reverse aging in engineered mice in every way imaginable. Now, nothing else has ever done this. Everything you've ever heard about curing aging or things that reverse aging, nothing has ever done what telomerase induction or telomerase gene therapy has been able to do in human cells. What I want to do is I want to show you, and I'm going to make it brief, a short video from Diane Sawyer interviewing a scientist that, using our technology, actually did this with mice.
And now, eternal youth. Is it in a cage around the corner? News tonight of a breakthrough for some pioneering mice. But we always wonder, what does a fountain of youth for rodents reveal for humans? Here's Sharon Alfonsi reporting.
I feel tremendous.
In the movie Cocoon, it's a swimming pool that turns back the clock for a group of senior citizens. But now, researchers have found a way not just to stop, but reverse the aging process. The key is something called a telomere. We all have them. They're the tips or caps of your chromosome, seen here in yellow. This is what it looks like in a young adult. But as you grow older, the telomeres become damaged and frayed, and as they stop working, we start aging, experiencing things like hearing and memory loss. Scientists took mice who were prematurely aged, added an enzyme, and essentially turned their telomeres back on. You can see it before the enzyme, after.
Their brain function improved, their fertility was restored. It was a remarkable reversal of the aging process.
Look at this picture. The mouse on the right has bad skin, gray hair, and is balding. But the one on the left had its telomeres flipped back on.
And you could see that essentially you now have a dark coat color, that the hair is restored, that the coat has a nice healthy sheen to it.
Even more dramatic, the change in brain size. This is before the mice had 75% of a normal brain, like a patient with severe Alzheimer's. But after the telomeres were reactivated, the brain returns to normal size.
Okay, I'm going to cut it there for lack of time. This is exciting. Essentially, they reversed aging in mice in every way imaginable, including possibly even reversing dementia, maybe Alzheimer's. I mean, it suggests that maybe Alzheimer's is not the loss of memory, but loss of access to the memory, because memories came back. But what I think is really important is that this could do this. This has the ability to actually reverse aging, as I show in the Betty White test. We just have to test it in humans. And the research costs to do this are not as expensive as the small molecule drugs, the gene induction stuff I was talking about. It's still pretty expensive. We spend about $200,000 a month on research developing this. And as I already mentioned, the treatment to actually cost, actually I didn't mention it yet, the cost to actually treat a person cost us, my company, $1 million per person per dose. So this is not exactly the goal that we want to achieve here, but it is going to provide, as I'll show you later, some proof of concept. What I want to say is that the first person's already been treated, and that's Liz Parrish. She will be treated, she'll be speaking later today. I'm sure she's going to give a very, very great talk, but she's already been treated. We have learned things from her treatment, stuff like that. So this is before we actually developed a company that I'm going to announce right now called Labella Gene Therapeutics. And what we're trying to do now is build on what Liz has done to try to see if we can actually come up with therapy that will actually turn somebody like Betty White back into a 25-year-old again. And those clinical studies are close to getting underway right now. It has taken us 2 years to complete the clinical protocols. They're about 140 pages each. We have clinical protocols now for treating Alzheimer's disease, critical limb ischemia, cardiomyopathy. We'll soon have one for treating demyelinating diseases such as multiple sclerosis and CDIP. And many more are coming too, but what I want to add is that, I didn't update this slide, we now have one that's just for aging. All these studies are looking at aging, but we also have ones just now for aging in general, and we're going to be measuring, even though I said we don't need biomarkers, over 150 different biomarkers during this study, biomarkers of aging. And then we have many more coming, and these are listed on clinicaltrials.gov if people want to find out more information about these studies. We're using a system, the gene delivery, that small little cell bubble is called AAV, for adeno-associated virus. That's what we're using. This gene delivery system has already been used in over 200 clinical studies and had very little, no serious side effects, except they did have some problems with immune sensitivity and some other things. I just want to make a point of that a lot of these studies, even though they were FDA approved, there were still serious errors that were made. Some people have died in some of these studies. There has been serious problems with side effects from immune responses and stuff like that. But it was mostly because the scientists, the medical doctors, and the FDA didn't read all the studies and weren't really prepared for this. But we have worked out on all these. We've got our clinical studies where we've solved this problem of immune sensitivity. Another problem is that 70% of us here have what are called neutralizing antibodies that will destroy the gene therapy before it ever has any effect on us. We've solved that problem too. Turns out the DNA that's inside the virus is immunogenic to some people. We've solved that too. And then the way it works out is that 0 people can be retreated. Now, Liz Parrish, maybe she could never be treated again because she's already been treated. But we have figured that one out too. So I'm looking forward to hopefully retreating Liz Parrish someday. And she's working on it herself too, so I should say. The funding, the issues that are going on right now include one funding. As I said, it costs $1 million per patient for us to treat the patient. I mean, when this becomes actually on the market, it's going to cost a lot more because the company doing this, Labella Gene Therapeutics, is going to want to make a profit. But this is the zero profit right here, because it costs about $1 million to do at the doses that we think are needed to actually make this work. Funding there, They're looking at investors, government funding, and pay to play. Okay, this is an option. Let me just discuss this. The FDA has recently approved under extraordinary circumstances, such as when the drug being tested has such a high price tag, i.e. $1 million a month or more, or per person or more, that the study can't be done unless the patients are allowed to pay for it themselves. It's not the first choice that Labella has, but Labella has been so far unsuccessful at finding somebody who would do the investing or other sources of funding to actually cover this. This is actually, you saw that the FDA clinical ruling regulation protocol right there. Okay, we also need to do preclinical studies. And there's already been a lot of third-party animal studies done, mostly with mice. One thing that I want to make really clear is that almost all these studies, they got the tools to do these studies from me and my company and previous companies that I've worked with. And that's because Sierra Sciences, my company, is a discovery company. We're not a testing company. So we don't do the testing. We rely on, we collaborate with other labs to do a lot of these testing. And so when you read about these are things that we can use now to support our preclinical studies. The problem is that most of them are doing it on mice, and I already said I don't really believe mice are a good model for studying human aging, or a lot of different diseases. So what I have done is I've done some investigating, and I found out that there's this close to human, it's a primate. It's the world's smallest primate, and here's a picture of it shown in Shaquille O'Neal's hand. It's called Madame Bertha's mouse lemur. It's only found in Madagascar. And what we are doing is we're working with people there to see if they can start doing some animal studies on this animal. And the only qualification is we don't believe in doing animal studies unless the animal benefits from the study. Okay, so that's why I'm hoping that we can do some studies. The animals will live longer, and then the data we get will help us live longer too. Okay, so that's stuff that hopefully we're trying to get underway. Locations for doing the studies, we're looking worldwide to have places for doing these studies. The first hospital has just been designated. It's a hospital, I can't even pronounce the name of it, near Bogota, Colombia. I just know it's state-of-the-art hospital, good doctors and stuff like that. I've been all over the world looking at hospitals. I've been very impressed with the quality of some of the hospitals in some of the countries that are really trying to become the go-to places to have clinical studies done. So this is one of the hospitals that we've just now selected. We're also been working very intensely on getting FDA applications completed. We've been actually working on, we're focusing mostly on something called RMAT, Regenerative Medicine Advanced Therapy, some new type of ruling that Donald Trump has created to allow people doing expensive gene therapy clinical studies. We're working on that. We started applying for this in September 2018, and we're still working on that. So the problem why things haven't been done, I said that Austin said we were going to treat our first person in November of 2019, hasn't been done because we're still waiting the funding. Funding has been the bottleneck in this whole thing, but it's now looking like the pay-to-play model is something that's actually going to bring us some funding, and we will be able to treat some people, and now I'm hoping it's going to be in early January. But our goal is to save lives. Sierra Sciences' goal, Labella's goal, 150,000 people die of aging every day. We don't want to keep delaying treatment to something that could possibly cure aging. So let me tell you who we are. The chief scientific lead for these studies is myself and my chief scientific officer, Lancer Brown. We do have some top scientific advisors that have had a lot of experience in gene therapy, Valder Arruda and David Scott. Our principal investigators, two really great doctors in the United States here, Dr. Sandra Kaufman, she was actually going to be here today and texted me this morning she's not going to be here, and Dr. Joseph Raphael. Now, you'll find that when you look at some of the studies on clinicaltrials.gov, the hospital will have identified somebody that works at their hospital to be the scientific lead or the principal investigator, but the true principal investigators are these two people. They're the ones that are going to be running no matter what's happening at the hospital, and so these are the people that I'm relying on. Very, very exceptional doctors. The president of Labella is Jeff Mathis. And what I want to say is that he lives in Manhattan, Kansas. And the reason why I'm mentioning this is because a lot of people think, therefore, Labella is in Manhattan, Kansas. But Labella is a virtual company. It's all over the world. It's owned by people from all over the world. It's just the president just happens to live in the United States in Manhattan, Kansas. So I would be amiss if I didn't talk about the fact that there are people around that are saying that what we're doing is going to give everybody cancer. And this has been a pain in my whatever, but I just want to discuss this a little bit. Some people think this is why we age. It's called antagonistic pleiotropy. People think that we age to keep us from getting cancer, and we age by shutting off the telomerase gene to keep us from getting cancer. But I think that couldn't be further from the truth. Now, does shutting off telomerase decrease the risk of cancer? Everything you hear is hearsay. There's absolutely no data at all showing that this is the case, just people talking about it. If it was true, it's failing because we're getting cancers at very high rates. Lots of people are getting cancers. I'm not going to go over all this. What I want to do is, there's a lot of reasons. Just say the lack of telomerase is actually far more likely to cause cancer than telomerase is. And if you want to find out in excruciating detail why I believe this, you can read my book, Telomere Lengthening. It has a chapter on telomerase causes cancer. It discusses all the reasons why everybody thinks, or some people think it does, and then proves why it doesn't. And if you don't like to read, there is a movie. I presented a talk in Tokyo where I spent like over half an hour, maybe 45 minutes, just talking about all the data. If you just go to my website, www.crsi.com, find that picture of an old truck, click on it, and start watching from 2525, you'll see every reason why I know that telomerase does not cause cancer. Okay, so in treating somebody, when do we treat somebody? This is true not just for our clinical study, but any clinical study, especially when it's first in human. Remember, first in human study is, these doctors have never done it before. Okay, these MDs do not practice on animals. Okay, somebody else does all the work in animals first, and then writes a protocol that the doctors then follow. So as a result, all those clinical studies I've said I've been involved in before, I've seen some pretty horrible mistakes made. Okay, because doctors, it's not, you can't blame the doctors, there's just mistakes. But I do think that being first in human in a clinical study, you are doing it at a risk. But when is the risk from no treatment greater than the risk from treatment? There's a lot of people, and this is why phase 1 studies are usually done on people that have a terminal disease, because their risks are very high. I personally believe we shouldn't be doing any studies on humans that we don't have any chance of saving. So in our clinical studies, anybody who's going to be in our clinical studies, or Labella's clinical studies, are in the studies because we think we might be able to save these people, even though they have a 0 chance without the treatment. So when the risk from no treatment is higher than the risk of treatment, I recommend that people get in these studies. Those include things like Alzheimer's and other different. Alzheimer's is 100% fatal unless you got killed by something else, run over by a bus or something before you died from the Alzheimer's. It's 100% fatal. So I think if you know somebody who has Alzheimer's, you should try to get that person into the study, especially if they can afford to do the pay-to-play model, or hopefully Labella will find funding elsewhere and be able to get people in at no charge. Just being old. I'm sure some of you here are 89 years old, but I'm sorry to inform you, but you have, even if you're a healthy 89-year-old, statistically you have an 11% chance of dying by 90. That's high risk, okay? I guarantee you, of all the risk I've ever seen in any clinical studies, there's never been an 11% chance of dying just from the treatment. So getting treated in some of these clinical studies is very important. The source for this data is down there on the bottom right, and I think that slide's included in your printouts. Okay, so the bottom line is a lot of times the most serious risk to the patient is not treating the patient, especially if you have Alzheimer's or extreme old age. you are better off treating than not treating. The person has a better chance of living longer. So with that in mind, I'm hoping right now the first patient will be expected to be treated in early 2020. I'm very disappointed to say that this has been delayed several times so far, and it's all because of funding. And people that have said that they were going to pay to play have kind of decided to hold off for a while. So nobody's been treated yet. But if you know somebody, if you yourself want to get treated, or if you know somebody that needs to be treated and can afford to do the pay-to-play model, or even if they can't and they want to sign up in hopes that some investor or government funding will come through to support this stuff, go to www.labellaGT, stands for labellagenetherapeutics.com, and sign up. So let me just talk one more thing. Why is this taking so long? I think I've already made it clear. It's the funding. We are doing the two different approaches, gene induction research and gene therapy research, to try to solve this problem. As I mentioned, the gene induction research costs us over $1 million a month. It's really hard to come up with that funding. The gene therapy costs about $200,000 a month. The treatment cost, if we came up with something that actually was potent enough by the gene induction protocol to reverse aging, it would cost us less than $1 per person to make the pill, whereas the gene therapy is costing over $1 million per patient, and I really doubt that that will ever become significantly less expensive than that before we actually come up with an alternative to gene therapy. But the point is, I look at this $1 million per patient as a great way to do proof of concept. Okay, so the scientist in me says, I hope people sign up because I want to get some kind of proof that it works. We're venturing into the unknown. I can say telomeres lengthen and telomere reverse aging, but we won't know until we actually test it on somebody. Just because it worked in mice doesn't mean it's going to work in humans. So proof of concept. We need a proof of concept, and hopefully we'll get that proof of concept from that. And when we get that proof of concept and Labella starts putting it on the market, Sierra Sciences will get a royalty, and we will be using all that royalty to invest in the gene induction research and come up with something that's less than $1 per patient. That's our goal. With the appropriate funding, I believe that we could have that pill in 1 to 3 years. It's just I've been saying that for 10 years because we haven't had enough funding to actually do this. So the sources of our funding right now come from companies like Isagenix, Defy Time, and Labella Gene Therapeutics. What my company really doesn't work with investors that much, but what we do is when we make discoveries, such as a way of measuring telomeres or some pill, we license it to some other company that then does the marketing, so we can just focus on discovering. We don't do testing or marketing. Then we get a royalty, and that's how we fund our research. And we do have a project that we think is going to generate a lot of funding. That's pets. We think treating pets with our treatments would be a cure for a source of funding. And we are actually developing something right now we're calling Telodash, named after my dog Dash. And we think this is just going to be something you sprinkle on your dog food or cat food that would then provide the therapies for our pets. The really big question is, who's going to market it? We are not marketers. We just do basic research. If you know anybody that's interested in the animal field and wants to take on marketing such a product, that door is wide open right now. So that's what I want to say. I hope to see you there when that happens. And it's my company logo. Thank you very much.